many awards like ICMR International Travel Awards and many more. Dr. Sahu, now I would like to request you to present your deliberation so that we will get some exciting information about your research. Dr. Sahu, please.
Is my screen visible?
Yes, sir, you are visible.
All right, thank you. Thanks to the RABS organizing committee, convener.
Audible and visible. Please continue.
Thank you. I am thankful to the organizing committee, the convener, patron, organizing secretary, and all for giving me this opportunity. So recently, I do not have a perfect lab, so I work myself because 6 months back I joined. So I started a small work on effect of cyclin D1 down regulation on breast cancer cells. So this is well known in the literature. So means my aim is to work in the context of tumor virus infection. So here I will be using a model tumor virus. That's the first identified tumor virus, Epstein-Barr virus. So here a little introduction about the cyclin D1. This is well-known regulator of cell cycle. The cyclin D1 level.
Dr. Sahu, please make it full screen.
I am a little bit sorry about this. I don't know. I made it like full screen, but somehow it is not coming.
It's okay, sir. Please continue.
All right. Yes, sorry about this one. So cyclin D1 regulation during the cell cycle. So we know the level of cyclin D1 is higher in G1 phase. Then it will go down in S phase, and if cell needs to proliferate, then it will go again high in the G2 phase. But if the level of cyclin D1 will not go down, then cell will go on continuously proliferating. So cyclin D1 degradation is essential for sometimes in the S phase. So here this is a regulation. Like cyclin D1 level.
Dr. Sahu, I would like to request you to on the slide show.
So it is there some point.
Slide show.
All right.
Slide show of your presentation.
Wait a minute. Can you see it now?
Yes, now it's clear.
Yes, sir.
Thank you very much. I am sorry about the little disturbance. So if the cyclin D1 level will go higher due to some extracellular signal, it will interact with cyclin dependent kinase 4 and 6, and a complex will form over here. Then this will causes the retinoblastoma phosphorylation. Then E2F family will be free, and they will release the cell from G1 to S phase. So G1 phase, the cyclin D1 level needs to be high, then it will go down in S phase and again increase in G2 phase. So here this is a cartoon representation of the cyclin D1 level during G1 phase high, S phase low, and even cell needs to go to the G2 phase, then it will again go high. Right? So in most of the cancer cell, including breast cancer cells, the cyclin D1 does not degrade. So it got stabilized. So here we hypothesize that if we silence the cyclin D1 and at the transcript level or degrade it somehow, then we can reduce the severity of the breast cancers. And earlier we have seen that the Epstein-Barr virus, this is a tumor virus. So if this virus is infected, in case of breast cancer patients, then it stabilizes the cyclin D1. So to address this one, and the initial objective is to investigate the cyclin D1 silencing on breast cancer cell in the absence of virus infection, and the impact of this silencing on apoptosis of breast cancer cell in the absence of Epstein-Barr virus infection, and these two objectives in the context of virus infection. So this is a flow for cell cycle analysis, the experimental procedure. Harvest the cell, harvest the breast cancer cell using trypsin, then permeabilize the cells fixing by paraformaldehyde and treating with a low concentration of detergent like Triton X100, then incubate, wash, and analyze the cell cycle. So here G1 phase, S phase, G2 phase, M phase, like that we do the cell cycle. So here we can see in the control situation the G0, G1, S and G2 phase, and in presence of cyclin D1 silencing. So here we can see that silencing of the cyclin D1 blocks the cell cycle in the G1 phase. So here the number of cells in the G1 is increasing. Another way of checking like how it affects the apoptosis. So here the
Hello, Dr. Sahu, you are frozen. Something wrong there. I think Pragya will give a brief introduction of the speaker, Dr. Alpha, and next they can continue the discussion.
Welcome you all. I would like to give introduction about Dr. Manmohan Arka. Dr. Manmohan Arka is presently working as an officer in Center for Biotechnology, Shiksha Anusandhan, University, Bhubaneswar. He is working on projects which focus on the interaction of nanomaterials with living systems to develop novel ways of using the nanoscale materials in precision aeronautics and advanced propulsion technology. He has done his PhD in National Institute of Technology, Department of Life Sciences, Brazil. He has got overseas postdoctoral fellowship award by SCRB India. Also, he has won patent on method for continuous production of silicon nanoparticles using bacillus thuringiensis. Now, I would like to request Dr. Arka to deliver his talk. Dr. Arka, you may please.
Sir, please unmute yourself, sir.
Am I audible now?
Yes, sir, you are audible, sir. Sir, call the-
Yes.
Yes, sir. You can go on full screen.
Good morning to all. So, I am Dr. Manoranjan Arakha, working as assistant professor at Center for Biotechnology of University, Bhubaneswar. So, today, my topic is biogenic particle from Punica L and evaluation of its antioxidant, antimicrobial, and antiviral activity. So, before going to the topic, so I would like to give you some brief introduction of nanotechnology. Okay, so during a conference at in 1959, so one scientist, Richard P. Freeman, so he covered a talk. There is plenty of room at the bottom. Okay, so in that talk, you would like to say if we could able to manipulate matter at atomic and molecular level, then there will be generates the new matter. So that new matter which will be generated will possess new okay. That matter we can tell those materials as nanoparticles or nanostructural materials. And we can tell these materials because these are very tiny materials having size in between 1 to 100 nanometer. And since these are very small, it possesses different advanced physical chemical properties like high surface to volume ratio, high surface free energy. Okay, so when these materials in case of biological field, or when these nanomaterials are dispersed inside the biological fluid, so generally it will come in contact with different biological macromolecules or biomolecules, so forming a nanobio interface. Okay, so let's see what is this nanobio interface. You see this picture. So you see in this picture, there is an interaction of a nanoparticle with a biological membrane. Okay, so the surface which is very common to nanoparticle and biological membrane is known as nanobio interface. Okay, so why this nanobio interface is so important? Because all physical chemical interactions between nanoparticle and biomolecule happen through this nanobio interface. Also, thermodynamical actions, kinetic actions, all things happen between nanoparticles and biomolecule. So, if you want to determine the fate of nanoparticles inside the biological environment. So you have to understand the interactions happening at nanobio interface. Okay? By understanding this interaction, you can easily determine the fate of nanoparticles inside the biology. So as a consequences of this interaction at nanobio interface, so sometimes nanoparticle will act as a biocatalyst. Also, intracellular uptake of nanoparticles happen, and sometimes nanoparticle may biocompatible to the cell, or sometimes it will show toxic effect. Also, inside the biological fluids, protein molecules also coat over the nanoparticle, forming protein corona, which have also different biological applications. Okay. So nowadays, antimicrobial resistance has become a global health concern. Okay, so you see, so like Center for Disease Control and Prevention have predicted that, so our world is going into post-antibiotic era, where antimicrobial resistance or resistant bacteria will be developed, which will cause different infections. Have predicted that people will die from bacterial infection. So that people are searching for alternate antimicrobial therapies which will combat this antimicrobial resistant bacteria. Okay. So in this context,
Manoranjan,
yes, sir.
slide show karo.
Sir, already କରିଛି site slow
Full screen, full screen.
full screen sir କରିଛି. Sir, already.
Okay, continue.
Sir, I think there is some network issue or something happening, but it is in full screen. Okay. So, nanoparticles having different physical chemical properties can be used as alternate antimicrobial therapy. Okay. So, these nanoparticles can be synthesized using different techniques, different process, like physical process, chemical process, and biological process. And nowadays, biological synthesis process has been gaining importance due to non-toxic or it is eco-friendly nature. Okay. So in this context, we are designing an experiment, or we have performed a biological synthesis of silver nanoparticles, okay, using pomegranate peel extract, because pomegranate peel has high, there are a presence of different flavonoid contents, protein molecules, which will synthesize silver nanoparticles. Okay. So briefly, in experimental process, briefly, we have synthesized silver nanoparticles using biological methods. Then we have characterized the silver nanoparticles using different techniques like UV visual spectroscopy, XRD, ATR, FTIR, FECM. And after that, we have studied the antimicrobial activity of silver nanoparticle using growth kinetic analysis, colony forming analysis, and activity H2 free rating of acidic acid. And also we have studied antibiotic activity using and macroprotective flat assay. Okay, so coming to results in this, so when you are adding silver nitrate, then there is color change. Okay, we have observed the color change, so which confirm initial synthesis of your silver nanoparticles. Okay, after that we have analyzed these nanoparticles using UV visual spectroscopy. In UV visual spectroscopy, when we analyze the nanoparticles, we got peak at 462 nanometer. Generally, silver nanoparticle, due to its surface plasmon resonance activity, it gives peak around 420 nanometer. So if you are getting peak at 420 or around 420 nanometer, then you can confirm that your nanoparticle is silver nanoparticle. Okay. So after this, we have also characterized the nanoparticles using a FTR analysis, sorry, XRD analysis. Okay. So in XRD analysis, you see the XRD spectra, where in XRD spectra we talk diffraction peaks. These sharp diffraction peaks show inform that so whatever nanoparticles we have synthesized, these nanoparticles are crystalline in nature. Also, we have also calculated the size of nanoparticle using this XRD data, and we got the size of nanoparticle to be 20 nanometer. So after that, we have characterized the synthesized nanoparticles using IR spectroscopy. In IR spectroscopy, we see functional groups present on the surface. Particles in IR spectroscopy. So we got a peak, you see, around 426 nanometer, which demonstrate that there is interaction between silver and oxygen or silver and silver. So if there is interaction between silver and silver or silver and oxygen, then you will get a peak, so below 800 wave number. So since we are getting peak at 526, so initially we confirmed that there is synthesis of silver nanoparticle. Moreover, we got peak at 1634. So this peak is due to amide 1 vibration of the protein. So we confirm that, so the protein molecules are there which synthesize the silver nanoparticles. Along with synthesis, these protein molecules are attached to the nanoparticle surface. So due to which we are getting the peak at 1634. Okay, so apart from this, other peaks we got which are due to different functional groups or flavonoid compounds present in the plant extract. So next we have seen the morphology of synthesized nanoparticles using FESM. So in electron microscopy, we got nanoparticles. Most of the nanoparticles are spherical in shape, and we got size and measured the size. The most of the nanoparticle size was varying from 40 to 80 nanometer. Okay, these were characterization of nanoparticles. After that, we have applied this nanoparticle for different biological studies. So initially, we have checked the antioxidant activity of the synthesized silver nanoparticles using DPPH antioxidant assay and H2O2 free radical scavenging assay. In DPPH assay, we have seen with increasing concentration of nanoparticle, so we got more antioxidant activity of the silver nanoparticles. Okay. Similarly, in H2O2 free radical scavenging assay, we got silver nanoparticles have the potential to scavenge the free radicals formed due to free radicals form, and it has antioxidant activity. So after that, we have checked the growth kinetics analysis to check the antimicrobial activity of silver nanoparticles. So we have taken two bacteria like Bacillus cereus and Vibrio cholera. So we have measured the antimicrobial activity, so using growth kinetic analysis, and we have seen, so you see the control has kinetics, and the control is increasing, the growth curve is well in case of control, but when we are increasing the concentration of silver nanoparticle starting from 10 microgram per ml to 500 microgram per ml, with increasing concentration of silver nanoparticle, the growth inhibition of the bacteria happen. Okay, and more growth inhibition happens after 25 microgram per ml in case of Bacillus cereus, and in case of Vibrio cholera, the concentration is little bit higher, like 50 microgram per ml. So, however, we have seen the synthesized silver nanoparticles have the potential to inhibit the growth of both gram-positive bacteria and gram-negative bacteria. So also the antimicrobial activity of the synthesized silver nanoparticles has been confirmed by studying the colony-forming unit quantification. Okay. Similarly, we have measured the colony-forming units of Bacillus cereus and Vibrio cholera. Then, with addition of silver nanoparticles, we observed that with increasing the silver nanoparticle concentration, the number of colonies decrease exponentially. Okay, so which confirm that silver nanoparticles has the antimicrobial activity. Similarly, we have checked the antibiophilic activity of silver nanoparticle against Staphylococcus aureus. See, the control has shown the biofilm activity because blood colonies are formed, but with increasing the concentration of silver nanoparticles, the biofilm formation decrease. The antibiofilm activity of silver nanoparticle was further confirmed using microtiter plate assay. So where we got the control has higher observancy showing higher biofilm formation activity, but with increasing concentration of silver nanoparticle, the biofilm formation activity of the bacteria decreased gradually with increasing concentration of silver nanoparticles starting from 25 microgram per ml to 500 microgram per ml. Okay, so although we are studying different mechanism, what is the mechanism behind the antimicrobial activity of different nanoparticles like silver nanoparticles? So here one mechanism I am showing. So we have studied what happens to the bacterial membrane upon interaction with the nanoparticle. You see, when AGNP is interacted with a bacteria, suppose Bacillus cereus. So in case of Bacillus cereus, we have checked the morphology of Bacillus cereus using FECM, and we got rod-shaped bacteria. But when AGNP was treated with this bacteria, then the membrane is disrupted upon treatment with silver nanoparticle. So we conclude that silver nanoparticle has the potential to disintegrate the membrane of the bacteria. So this is overall schematic diagram, whatever work we have done. So like this, the biogenic synthesis of silver nanoparticle. So we have added silver nitrate to the plant extract, then silver nanoparticle was formed. So after that, we have checked antimicrobial activity, which is killing the bacteria, which has strong antimicrobial activity, also strong antibiofilm activity. So as a conclusion, so we got the peel extract of Punica grantum L or pomegranate peel extract has the potential to reduce silver ion to silver nanoparticles. And the silver nanoparticles, whatever synthesized, were characterized using different biophysical techniques like visual spectroscopy, XRD, ATR-FTR, FECM analysis. And one more important information we got from ATR-FTR analysis that whatever protein molecules are there in the plant extract, these protein molecules help in the reduction of silver ion to silver nanoparticles. Then the biological synthesized silver nanoparticles show strong antimicrobial activity and antibiofilm activity. So coming to acknowledgement, I would like to acknowledge my PhD supervisor, Dr. Suman Jha, then Professor Sanghamitra Naik, HOD CBT, Shwa University, and UGC for financial support, and my PhD scholar, Banisri Sao and Lipsalina Panigrai, who has executed the experiment. Okay, thank you.
Manoranjan,
Yes, sir.
very nice work. So this work we publish.
Yes, sir, already published.
Thank you. Thank you
Thank
very
you,
much
sir.
for your nice presentation.
Thank you, sir, for giving me this opportunity.
If no questions from audience side, then you can thank you once again for the nice presentation.
sir.
Hopefully we'll meet soon. Thank you, sir.
Thank you very much, sir. Thank you.
Dr. Sahu,
Yes, sir.
you can now continue your
Thank you, sir.
presentation. Sorry for the inconvenience. Okay,
I am
how things
actually
move.
sorry about. Sir, is my screen visible?
Yes, sir, it is
Yes,
visible.
sir. You can continue.
Yes, sir. So here is a simple experiment for the cell cycle using propydium iodide staining. So in the control siRNA, we can see this is like G0, G1, S and G2 phase. when the cyclin D1 is silenced, then the G1 phase is increasing. So it is arresting the cell in the G1 phase if we will silence the cyclin D1. Another experiment was done in the presence of Epstein-Barr virus infection. So if we will infect EBV infection and repeat the same experiment, then it was releasing the cell cycle. So it cannot arrest the cell cycle in the G1 phase. So that experiment I have not shown over here. Or another experiment on the early apoptosis. This is a well-known mechanism in the normal cell. This is bilayer. In the early apoptotic stage, the phosphatidylserine from the inner leaflet goes to the outer leaflet, and in the outer leaflet, outer membrane, we can stain with annexin 5 and check the early apoptotic cells. So this was a very good experiment. This is like percentage of apoptosis in control and cyclin D1 siRNA. So here a little increase in apoptosis. Apoptotic cells are increasing. So here only early apoptotic cell has been shown. So our aim is to work on the early apoptotic cells. So this is early apoptotic. These are some of the early apoptotic cells. When we can see here, like this is control when there is down regulation of cyclin D1 by siRNA, then there is a early apoptosis. But the same thing in presence of Epstein-Barr virus infection, the apoptotic cells we are not getting. It means the virus is inhibiting the cell to go apoptosis, which is mediated through cyclin D1. So this is a very nice experiment I am saying, because we are getting some information that EBV means Epstein-Barr virus. These viruses are like latent virus which made in the lab and infected. So some of these latent viral proteins might be doing something with the cyclin D1. So I am thinking like there are like 10 proteins, viral proteins in the latent phase. Epstein-Barr virus nuclear antigen 1, 2, 3A, 3B, 3C. Ibna leader protein, latent membrane protein 1, 2A, 2B. Like that, 10 viral proteins are there. We have cloned all those viral proteins and trying to check which viral proteins is playing role to manipulate the cyclin D1. So that experiment is going on. That is one part. In another work, like the cyclin D1, there is a single nucleotide polymorphism that is at the position 870 at the junction of exon 4 and exon 5. So if at this position 870 position, if G will change to A, then if there is a GG, then this is like cyclin D1A, and if this is AA, then that is cyclin D1B. So there is three conditions: GG, that is the homozygous dominant, GA heterozygous, and and AA homozygous recessive. So cyclin D1B form, or we can say like at 87 position, if it will be AA in place of GG, then it has been observed like D1B form observed in most of the cancers. So there are like more than 100 reports, wet lab epidemiological reports that cyclin D1B is associated with multiple cancer, also in breast cancer. But in the epidemiological study, there are some intriguing reports. Some reports are saying like if this polymorphism is happening, then it is associated with cancer, and it is some other reports are saying it is not associated with cancer. So in a epidemiological approach, like whatever primary data is available, we did a analysis by on comprehensive meta-analysis. So this is to show from a report, like here you can see like this is a PCR RABS of CCND1 gene. So if there is like 167 base pair, that is GG. If that is 145 base pair, this is AA. And if both the bands are there, then this is GA. So like that, homozygous dominant, heterozygous, and homozygous This is it for GG, GA, and AA. So like that, PCR RFLP sequencing people has done in multiple country and such like association of this polymorphism with breast cancer or not association. Also in some other female reproductive cancer has been done. So we like did if we follow the Prisma guideline that has been published in a paper like PLOS Genetics, and search some article in the literature and found 27 article eligible for female reproductive cancer which are available in the database. So this is the comprehensive meta-analysis interface. And this is in the search criteria. We review the literature, study the abstract, and find all these articles. So these are on breast cancer, and they are on cervical cancer. So these two are female reproductive cancer has been targeted. So it has been analyzed in 5 different models, model 1, 2, 3, 4, 5. And this is a forest plot only I am saying. There are multiple other analysis, but from here we can find some conclusion. So when we analyze some of the study, for example, this study in 2005, we can see here the odds ratio is more than 1. It means this polymorphism, G2A polymorphism at cyclin D, it is associated with cancer. And here we can see p-value less than 0.05. If this is more than 1, odds ratio is more than 1, then it is associated with development of cancer. Another study here we can see this is less than 1. So it is saying this polymorphism like G2A polymorphism is giving protection. So there are like that we got 27 articles in the literature, and with multiple number of cases and controls. So we did an analysis. This is called the allele contrast model, and we combined based on primary data we studied. So you want to see like what is happening in the real term, like in the epidemiological database. So when we combine the result, we saw it looks like this model is giving a protection, like G2A is giving a protection for the development of cancer. But this is trend of we will say trend of protection, but this is not true because the p-value is more than 0.05, more than 0.05. So it means this is not associated. So in this model we did not get the association. This is the called allele contrast model, and this is the forest plot analysis. Also we did other analysis like funnel plot analysis, sensitivity plot analysis, and many other statistical analysis we did. So in all those cases, it did not give any association. Like that, it looks like similar figure in the model 1. Here this is like model 2. This is like AA versus GG comparison at the cyclin D G870 position. And here also we did not find any association. We can see here like the p-value is more than 0.48. So although it is giving a trend of protection, but this is not significant due to this higher p-value. Here also we can see this is giving a trend of association, but not true due to higher value of p-value. So like that, model 4, model 5, like that we did. So from this we can say actually we analyze different sample all over the world, and it will total the sample. Then this was like 14,886 and patients and 16,092 control. So this is a very high number. We did a power analysis for this. How true is our study? Then in this case, power analysis that is like more than 99% means this is the study is highly significant. And we are saying we did not find any association between cyclin D1 polymorphism and the cancer. So so from the in conclusion, I can say like in the context of virus infection on some of the viral proteins, I don't know right now know which one is doing something to the cyclin D1. So that cyclin D1 level may be stabilized, and it is allowing the cell to proliferate in an uncontrolled manner. So it is contributing towards the cancer, but in the wet lab work, like in the cell line models. But if we will see in the epidemiological field, one of these polymorphisms, cyclin D1 G8702A, although many reports are there that this is associated with the development of cancer, but in our comprehensive meta-analysis, we did not find any association. Thank you all for listening to my talk. If any question I would like to like to take, and I will be happy to take question. Thank you the organizing committee and all audience for keeping patience and listening to me.
Thank you, Dr. Sahu. Nice talk. So I have a couple of questions. So
Yes, sir.
what is the significance of, preferential infectivity of that virus to breast cancer? What is the preference?
Preference. Okay. In the
Not audible.
sorry, sir. In the literature, like in the literature, we get like latent 1 infection more than 90% population of the breast cancer, but it switched to latency 3 when the severity is high. So more than 90% breast cancer patients are like positive with latent infection, like latency 1. It maintains actually a long latency. So, like based on 10 viral proteins. So in the latency
And next
1.
question is,
Right. Yes, sir.
you studied polymorphism. So have you studied that mutation, single point mutation, effect on cell cycle progression?
In the cell cycle progression, I have not checked, but people earlier have reported like if this polymorphism is there, then the cyclin D1B form that will accumulate and release the cell cycle from G1 to S. If cell cycle needs to arrest in G1, but it is able to release the cell cycle from G1 to S. So that is there in the report.
Okay, thank you.
Welcome.
Thank you, Dr. Sahu, for your nice presentation. Hopefully
Welcome,
all
sir.
of you have benefited from your deliberations, and we will definitely continue to expect a lot from you in future. Thanks once
Thank you.
again.
Yeah, welcome.
Professor of the Cornell University. We will be talking on conservation, restoration, and phytochemical analysis of mangroves of Sapta District. And I think there is no introduction to Radindra. He is a well-known face. He is an on-ground boy. I know him for a pretty long time. He has been doing good work, and he is known as a very efficient professor teaching in different dimensions of botany or plant science. Now, in this introduction, I have to hand over to Radindra Kumar Naik, but it will be a great help if you limit your presentation to 15 minutes, Radindra, to the maximum, if you are listening to us. Professor Radindra, if you are listening to us, I request you to limit your presentation to 15 minutes maximum. Due to paucity of time, we have lot many presenters here. We have to accommodate people next. So this is my request on behalf of the organization. Thank you.
Okay. Thank you, Jitu bhai. So, will I give my presentation now?
Yes, sir. Namaskar, sir.
Okay, namaskar, Sujit. Okay. So, in fact, it is a matter of great pleasure on my part. Chinu. Good morning, Chinmaya. Am I audible? Sujit,
Yes, sir,
am I
you are
audible?
Yes, sir.
Yes. Okay. So good morning, everybody. My esteemed elder brother Jitendra Bhai and my dear student Sujit, my younger brother Chinmoy, and esteemed principal, my friend Gopal Haldar, convener Dr. Kichak Kumar Jana, organizing secretary Pragyan Paramita Naik, and my colleagues, ex-colleague Braj Kishore, and other resource persons. It is in fact a matter of great pleasure on my part to be associated with this international seminar. In fact, I had planned to go to Vikram College, which is a premier college, but due to some health problem, I could not go. And now, in fact, I'm just going to show the PPT. Just a minute. So is this screen visible?
Yes, Professor Anindra. They are visible and audible.
Okay. So my topic of presentation is conservation, restoration, and phytochemical analysis of mangroves of Sartha estuary. So, in fact, I have just to present some aspects of these present status of mangroves. So before coming to the mangroves of Sartha estuary, let me just give you a broad idea because some students are also present there. So in fact, these mangroves are important natural resources, which includes biotic resources as well as abiotic resources. And God has given us resources on the surface of the earth, these biotic resources, and under the surface of the earth, these abiotic resources. So we should have a proper management of these resources so that we can, or our resources can sustain for future generation. And these biotic resources includes plant resources, animal resources, and microbial resources. And these plant resources include these phyto resources, which are the source of food, clothing, shelter, herbal medicines, and they have also got other economic importance and environmental significance. So far as the state of Odisha is concerned, we have reached phytoresources or plant resources because of our geographical locations. And we have mountain ecosystem as well as coastal ecosystem. And some of these important biotic resources or phytoresources includes our Similpal Biosphere Reserve, Mahendragiri resources, which has been recently declared as biodiversity heritage site, Gandhamardan hill ranges. These are some of the important mountain ecosystem in our state. And so far as the wetland ecosystem is concerned, we have this Chilka Lake, mangroves of Bhitarkanika National Park, Mahanadi Delta, Dhamra estuary, and of course Sartha estuary. And we know that these phytoresources constitute a significant portion of our biodiversity. And when we talk about their present status, conservation, and restoration, we come across this biodiversity conservation. And you know that this biodiversity of the state of Odisha, as well as the other part of the world, is now subjected to great degrees of threat. That includes your population explosion, industrialization, urbanizations, and a number of factors, and above all the climate change. And we know that this climate change, which is mostly due to the various activities of the man, have caused much threat to the biodiversity. It has given rise to environmental pollution, global warming, which affects the status of our biodiversity. And we know that these biodiversity are very much essential for our environmental protection. And I will just give a broad idea about the role of these mangroves. And mangroves, they play a significant role in the protection of our coastal environment. And because of the loss of mangroves, mostly in the state of Orissa, we have become the victim of many natural disasters. We can recall the super cyclone of 1999 and the recently Fani, which have got much devastations in the state of Orissa. And it is because of the loss of mangroves in the coastal areas. These mangroves, mostly for the students I'm speaking, have certain special ecological adaptations like pneumatophores, viviparous germinations, and presence of salt excretory glands. Because of these adaptations, they can sustain in the coastal areas. So these are some of the adaptations of mangroves. And because of these mangroves adaptations, mangroves can sustain in very top environmental conditions in the coastal areas. These mangroves, they have got much economic importance and also environmental significance. And we know that very often due to this global climatic change, because of this greenhouse effect, there is a melting of the ice cap, which also have become resulted in the fluctuation of salinity level of the mangroves, which affects the status of the mangroves. And this mangroves of the state of Orissa is also subjected to this global climatic change. And because of this climatic change, the status of the mangroves in the state of Orissa is much alarming. That we have realized in the last super cyclone of October 99, and we have seen that there was a disastrous situation in the regions of the Mahanadi Delta, where the mangroves are present in most denuded conditions, in the Kendrapara district, in the Jagatsinghpur district. Of course, the loss was comparatively much less in the Bhitarkanika area because it is a national forest. And mostly, I'll just give a broad idea about these mangroves found in different locations of the state of Orissa, which mostly includes the mangroves of Bhitarkanika, Mahanadi Delta, Devi Estuary, Dhamra Estuary, Sartha Estuary, and Talasari Vichitrapur area. These mangroves are mostly restricted to these Puri district, Jagatsinghpur district, Kendrapara district, Bhadrak, and Baleswar district. So far as the mangroves of the Bhitarkanika is concerned, the status is much better here, and it is famous for the saltwater crocodiles because it is a national park and it is well protected. And this status is here is much better because much thrust is given on its conservation. So I'm just showing some pictures that I have visited in these areas. These are the saltwater crocodiles, and these are the mangrove species of this Rhizophora apiculata, having the steel tooth system, which helps them to resist the effects of the cyclones. And the status of the mangroves of the Mahanadi Delta is much alarming. That we have realized in the 19th super cyclone. I have conducted some studies in the mangroves of this Mahanadi Delta since last 20 years in the areas of this Paradip, Batighar, Bukitola Island, Jambu Island, etc. These are the pictures I have taken 20 years back. This is the Paradip Hospital Limited, and here these are the phoenix species to which we call it a hental, which were found in the denuded conditions. Of course, now the vegetation has been little. This is the Bukitola Island, and it is an ideal site for the in situ conservation of the mangroves because this area is less interfered because of its geographical isolation. Government is planning to have certain ecotourism to these areas, but care should be taken for the conservation of the mangroves in these areas, which is an ideal site for in situ conservation because of its geographical locations. This picture I have taken at the time of super cyclone in the Hukitala Island. Yes, this shows the status of the mangroves in the Dhamra estuary. In the just December first week, I had been a visit with my research class, Avinash and Akash, and some other scholars. Here you can see the prawn culture in the Dhamra estuary, which is also a threat to our mangrove ecosystem. Just near the Dhamra port, we know that once upon a time there were mangroves in this area. But in that we should take steps for the restoration in some of these areas. No doubt, it is a question of bread and butter for some people, but at the same time, we should go for the restoration of mangroves for our future existence. Here, I have taken a photograph with an old man who has got much idea about the traditional uses of mangroves. Recently, there was the Odisha Environment Congress, which are in the Koraput, which are mostly based upon these indigenous knowledge. And this man have revealed some of the medicinal uses of mangroves. One species is called Exocaria, which is a very sticky substance. It has milky fluid. And he told that, sir, the milky fluid of this sticky substance can be easily removed with the help of another mangrove species, that is Sweda, that is an herbaceous species. And it has also medicinal importance. So my point is, some of the traditional knowledge, mostly about the medicinal properties of these plants, should be authenticated, which also we have seen in our future experiments, and which will be very useful for the bioprospecting. And this traditional knowledge should be preserved for the compilation of this traditional knowledge digital library. And now I'll give some emphasis on the mangroves of Sartha estuary. I had been to this area for some times, and I have observed some peculiar features there. This 19 super cyclone has much impact on the status of the mangroves of Sartha estuary. This is the Abyssinia species of the Sartha estuary. You can observe some plants are being caught, and here this is the Rhizophora micronata species. And during my study with my research scholars, we have come across 24 species of mangroves and their associates, which have got much economic importance. Some of them are having timber values, some have fuels, and some have medicinal properties. And this phytochemical analysis of some species, Soneraceae cassiolaris, Acanthus illicifolius, and Abyssinia alba, Abyssinia marina, and Rhizophora apiculata, reveals the presence of some of the phytochemicals like alkaloids, tannins, flavonoids, phenols, saponins, etc., which reveals their medicinal properties. That studies, of course, is in the preliminary stage, and subsequently we can have these antimicrobial activities, which is also being continued by some of my scholars, which reveals that these mangrove species have got much medicinal importance. And we know that these mangrove species in many areas is subject to severe biotic pressures. And in the Sartha estuary, we have come across a peculiar thing. These are the species of Abyssinia species and some other species. You can see the steel tooth system. And this slide shows a peculiar feature with Dr. Ashok Panigrahi, who was a former reader in Fakirmun Autonomous College. He is now associated with an international organization, Abdania, whose chief is Vandana Shiva. The places where we are standing, once upon a time, there are the creeks. But this 99 supercyclone has filled these creeks with sands. In the present time, we are also observing that even within the span of 3 to 4 years, these areas are now being covered with sands. So unless these sands are removed, gradually this mangrove forest of this Sartha estuary, it is decreasing because of the accumulation of sands. And we know these mangroves, they need muddy soils. So unless steps are taken, time will come when this forest will be vanished from these areas. This picture shows the restoration of mangroves in the Sartha estuaries. The local people, they were involved with this organization, international NGO, and they are planting the hypocotyl mangroves in these areas. And now I am coming to the concluding part of my topic. What we have observed that the status of the mangroves in the Sartha estuary, in the Mahanadi Delta, and other parts of the Orissa is much alarming. And these denuded status of the mangroves have increased the intensity of these mostly oceanic cyclones. If there were more species of the mangroves in these areas, in fact, the effect of these cyclones would have become less. I can tell you one thing, that the Chilka Lake, once upon a time, there were also mangroves. Now there are very few mangroves associates like these Acanthus silicifolius, and few species are there. And it has been authenticated by paleostratigraphical studies of the scientists of Birbal Sahani Institute of Paleobotany. They have excavated the soil, and that soil contains the pollen grains of some mangrove species. That reveals that once upon a time there were mangroves, but now these areas are being converted into the pisciculture. And a similar is also situation of mangroves on Sartha estuaries. In many areas, now there is prawn culture, and it is being represented by scanty vegetations. That indicates that once upon a time there were mangroves, but now it is in denuded condition. So these mangrove species, it should be conserved. Further phytochemical analysis should be carried out, and antimicrobial activities should also be carried out, which will be very useful for their bioprospecting. So it is the high time that these mangroves should be restored in the denuded areas, and present mangroves should be conserved, which will be very useful for the protection of our environment. Okay. So thank you for giving me an opportunity for this presentation. Okay.
Thank you, Dr. Anindra.
Thank you, Jitendra sir.
Now, any questions from the audience?
Okay.
Okay, you have touched many fundamentals. I think our
Yeah.
honors and postgraduate students will be definitely benefited.
Sir.
I myself don't have any question now to raise. If
Okay.
from audience no questions, then you can continue with the next presentation. Thanks once again, Professor Anindra.
Thank you, sir. Thank you, Jitendra bhai. Okay.
and people are really concerned about the biodiversity around us, how to conserve, and what we know today. Numbers numerically is very place. In the taxonomies, and another things, many of the macro start from the higher plants or animal comparatively easier than to identify the microbes or particularly microalgae. So I will touch up the taxonomies, what they are doing, what where, and what we can do with the traditional things and the new technology being used to come to a particular group of organisms which is really difficult to identify. How to come to the taxonomic conclusions. Please next. Biodiversity, diversity is reflected on varieties, maybe it comes from ecology, changes as per the condition changes, also reproduction type, and it's very from place to place. Now people are thinking geographical diversity. It includes all organisms. Place to change one place to another place. Also reflect sometimes phenotypically, but why people don't understand sometimes how they are varied in their genetically. So if you see the next one, it is also one type of algae group, diatom. Usually, what is the importance of algae? You know they don't need to describe. It's a very, very interesting organism, just like a simple plant. Do something like producing oxygens, but very importantly, the 90% of the oxygens coming from our biological things during evolution is come from the phytoplankton, the microorganism. And interestingly, every third breath we are taking, the oxygen, which was the process of worship, we are taking oxygen, it's come from the diatom, which is found basically usually in the marine environment, also in freshwater environment. So we must think about it. Next, please. Another group of algae, usually it is very close to the plant, green algae, to understand how the evolution of land plants come. Start from cyanobacteria to algae, then followed by finally the flowering plant. But interestingly, algae is very, very similar to the plant because of their pigment P, chlorophyll A and P. And if you go for analyzing most of the algae, the phytoplankton, they are highly diverse in the freshwater environment rather than the marine environment. So how we can say it is highly diverse? Because most of them microalgae, microscopic, and all looks alike. So sometimes it's come with the species, varieties, or so confusing. So if you go together, looking under the microscope, thinking differently of the structure, and particularly rDNA, ribosomal RNA analysis, 18S, if it is eukaryotes, eukaryotic algae, if it's cyanobacteria, usually you do the 16S RNA. Okay, so if you go for origin, now algae also coming under interesting group that is knowledge phyta, including the bryophytes, pteridophytes, and the angiosperm. Because few group of algae, particularly green algae, charophytes now, they are part of the streptophytes. Next please. So these are all the changes. Why I am thinking most of the information, particularly because of region India, most of the come molecular studies on cyanobacteria. Because wonderful environment to study throughout the India, you will find different types of cyanobacteria, blue green algae. But green algae, because the eukaryotes, it is very difficult to cultivate, whatever take from the environment. If you wanted to cultivate, it's never like Senadesum, Florida, Rhamnomonas. They are often very difficult to handle because each organism you have to utilize a particular medium. So that's why the less information found in the green algae, green microalgae compared to the cyanobacteria. Next please. Then again, usually now people are afraid to do taxonomy, particularly young people, because it is tedious. You have to go to field, you have to take, you have to process the sample, you have to preserve, you have to see on the microscope, it is not tedious. But you take the sample, then the DNA, then go for sequencing, but you cannot publish. But every day, if you do something, taxonomy, particular ecology, or looks different, we can say it's a new from the particular area or particular ecosystem. If you go deeper and deeper, like structure molecular, definitely come to a conclusion. If you are combined together, marriage in between the traditional taxonomy and the modern taxonomy, like molecular taxonomy. So now, if you wanted to do taxonomy, if you do normal things, sometimes people not accept your new hypothesis, new thinking, no new critical things. You are not evidently provide sufficient evidence like molecular structure. Qualification means you have to do the taxonomy, like microorganisms for the microscope, then go for the ultrastructure like scanning or TEM, or you can go for again do some molecular like 8 days, 16 days, or another one marker is ITS, internal translation. So my work is combination of all. I'm looking at taking the organism complete, looking at a microscope, then doing the structures like having this scanning electron microscope, then we are analyzing different molecular. Next, please. Interestingly, you see the same organism cholesterol looks one form, one species, then another is different species. Till date, 47 species, but out of 47, clearly define only 20. The 20 fledges, these are all species. Other are small varieties, it's very, very confusing. Next, you see, this is the particular species here. According to the cell structure, different type of form varieties, around 47 out of 20. So if you look at the next please. So this is confusing. Shall we decide as per the cell varieties or spine? So many phenotypical characters come. Interestingly, some of the organisms growing in this field, their morphology itself, they are overlapping. Cultivate in the media, morphology completely different. Can you spell different species? Because you are looking only having photographs and microscope, looking a little bit different. You can say this is some species. But when cultivating, morphology different. Can you say these two different species? Then they come, you have to think critical. Hypothesis come. You must see the other ways. So this is the trend. If you wanted to go supplements your existing morphological data, go for molecular. Like you can go for long subunit like 18 subunit like 18 years, long unit is 26 or 28 reversible RNA, or 18 years RNA small subunit. Maybe you can go for the filter. But it only have to go to the some of this will not reflect all because this macro size around 2000 to 20,000 best pairs. If you go to RBCL, macro size is less, like around 200 to 100 to 200, maybe less than that. If you find any best pair chain 2 or 3, you can say the different species because ribosomal are also another is a independent chloroplast present in the plants or so you can go for mitochondria particularly, I think all animal species, that is also independent genome, that's conserved genome. If any changes found, you can go for the species level. Another interesting one, ITS 2. ITS 1 and ITS 2. ITS 2 give any, we know that AT and GC. If it GC changes with GT, it is known as the MSCBC. One part of these only going to G. Due to any type of condition, like climate change or ecological system, adaptation. So if you go for that one, like this 12 with all data, you can conclude. If any found any difference with the reflective period, then it can be conclusion new species or there is a changes going on, maybe you can expect a new species. Next, please. So this we go for the 18S RNA analysis because you know that 18RS is the conserved DNA marker. And see particularly this largest database. Go for any type of organism. 18S RNA. You can compare with your existing nuclear ribosomal DNA and what cluster organism. That is like IPS2 also. This is the thing. You change to GC, complete base pair change. If any one base pair change in IPS2, either 3 or 2 link, then you can say this. know that species simply they are looking morphological cell because are we growing in water usually. Usually also they follow asexual or sexual reproduction. But if any changes, one CPC changes, they are completely reproductively uncompatible. Sexual. So if one AU or GC changes, then it's obviously is completely western. Sometimes AU may be changed to GU. It's only A changes. One base pairs. Also, if you count 3 or 4 MCVC, that you can say relate to a another species of evolution. Next, please. Like this, we go for different type of informatics tools, predicting the secondary structure. Next, please. Next. This approach we adopt, taking organism, looking under the microscope, cultivate, again, go for checking on microscope register, then DNA extraction, then go for the sequencing, then go for secondary structural analysis. At the same time, also doing those are interesting work of type go for the scanning rate. What you can change on in the changes during the initial collection and also under cultivations. So this is the usual to go to So then we always take the sample, we kept one is representative sample with preservative. And we all kept without any preservative for a solution. Immediately we check under the microscope and select a single cell, not all the cells. Usually we go sticking method and serialization, but handling eukaryotes, particularly the green algae, very tough. That's every week or 15 days of 1 month. Dr. Chennai, do you have chlorella? Do you have sanitasium? Do you have nanochlorosis? It becomes very difficult to handle. Every time you will get, you are not get the desired organism. Instead of chlorella, you will get the sanitasium. Okay, very difficult to handle. Always we do the particular single cell isolation or repeated subculture, then we do the usually not happening simultaneously. We have more than 200 sarcolasm I have. Out of them, around 70 green algae in our laboratory. And students are working. All are isolated from Odisha or West Bengal. So, not only we are looking at a microscope, also isolating DNA isolation. Then we go for a marker analysis. Then finally, depositing in the NCBI database for the gene magnification. So you have to represent. So otherwise you cannot define. But our gene comparison after publication will disclose, not before that. I never disclose because anybody can take it and manipulate it at the mind of the plan. It's very easy because we are handling the computers. So you see, if somebody says not psychologist, all will understand. But within that, we have seven species. Next, how you confirm? Next. All are stunning. Now you can see this is the arrangement of synovium, how it looks. But each one has even individual characteristics. If you look on a normal microscope, phase contrast to light microscope, not different. So we compare the light microscopy with this stunning one. What is the difference? Next. Yes. So finally we compare all the things, nothing new. Whatever published record is there, last 100, 200 years, we have not collected any new things. So it is disaster. After doing all things, 3 years, 4 years, students have nothing in hand. No. Then you have to think critically. What next or what is new in your sample? During collection, during cultivation. So you found this is interesting. It is just normal cell collected from the sample from the nature. And we cultivate changes like this. And we compare with this scanning, not much changes. Sometimes aggregated, sometimes separated. But whatever information in previously Heliotona out of Coelastrum, new genus is described because this arrangement, synovial arrangements, is none of the people describe about this. Even people say the Heliotona reticulatum, our species is completely different in outer structure, also in active to secondary structure. Next, please. So we adopt different type of, usually I see sequencing बहुत download करे छी 300 और 400 base pair थी. If see 1800 base pair को marker, if you see only 400 base pair, can you decide what it is? Very difficult to speak. So we find out, first we do 18 SLA गोटे कोड universal देइछु दिटा marker. Okay, पढ़े असला genome marker हमें amplify कर दो. तब जो genome हमें marker टा करे पूरे another 6 next is 6 primary to go for the sequencing PCR. We got quantic sequence around 2000 to 3000 base pairs. 500 both sides we detected. And next middle one we go for the quantic and go for the hallucinating analysis. Yes. So we also compare with RAPD analysis using different markers, primers. Next. So we combine all sequence together, then make a single sequence. So each one around 200-300 base pair. So if it's 6 primer PI being used, 800 base pair of quantic sequence will come. So this is the difference. If suppose 1800 base pairs of 180 ribosomal RNA is there, if you found only 2 base pair changes, you see 1802, very less. You cannot decide it is a new organism, it will look differently. You cannot merge up, you can merge up. The phenotypically different accounts, genetically different by the. So this is 18x. Then next. Then 16x. In 6, I chose to only 200 base pairs markers. So out of them, if it is hand 3, finger 3 of the structure, any 2 will be changes. Both phenotypically looks alike, they are more prototypically compatible. In 2002, there was what has been published, not only the algae, published from the plant, published from the bryophytes, different. We found anything changes in their secondary structure ideas to any 1 or 2 base pairs out of 400 samples, they are prototypically completely uncompatible. So our findings out of these 2 interesting structures, even they are coming under the reticulatum, they are genetically also different. The evidence from the ultrastructure. Also, they are coming the domain of variant in the reticulum. I cannot change the genus because it is only the deviation of the phenotypic and little base pair in the ITS2. So that's why we rename our sample, which is collected from Nicopra Bhubaneswar. Another sample also Bhubaneswar plant. 2 from the Bhubaneswar. So we make it headed on a next list. So this is the latest evidence. This is what changes here. This is what changes here. Every figure there is changes in our sample. So around 3 CBC and 1 hemi-CBC or 1 hemi-CBC, 3 hemi-CBC found in our sample. Next please. So finally concludes whatever information we will put this. So we renamed it Heredona. That's for our new name of Odisha. So I represent all over the world. Odisha is not Odisha, it is Odisha. So I was thinking I will dedicate to my Professor Adhikari. But here is also another 2-3 names in Professor Adhikari name like cell bacteria. So I name it Heredona odiscensis. Okay. So this is a little finding. It is going to be published in a journal in this technologies without much you cannot work. Thanks to our funding agency. Next please. All my teams directly have to carry out the work. And because of training of molecular technology, we always constantly support what to next, how we can be thinking, what is the hypothesis, how we can think critically. If everything is out of that, how can you differentiate yourself, your data? You cannot throw out the data. Thank you. And very much thankful to organizer giving I think I mean sorry. College, but wonderful arrangement. Thanks to our principal sir.
Thank you, Murtuja, for your talk.
All
Thank you,
team of
Murtuja, for your
biology,
talk.
biology and botany, and all students actively working everything. and I enjoy cultural program very much. So it is very, very pleasure and a good moment for me to come here. Thank you very much.
No questions, then we can continue the next speaker.
In taxonomy, I think no question.
Too many. The problem is there are too many questions. We are confused which way to air our questions.
Thank you very much.
I isolate the cards. Then I use specific primer. Then to only the 18S and IPS. IPS 1 and 5.8. Then after applications, we go for funding, combining all the primers, whatever they come from the application. Then we go for the secondary structure of IPS 2. Then just as specific. Out of that, GP to the required component is complete. 8 theta GC but 8 theta GT then I'm going to part of the DNA state already changes. Or if it changes not, then it will that they are not compatible. Thank you.
Actually, I have one but these are much better to base care. They are
Non-compatible. This is published in 2002, followed by 2006, followed by 2011. How we can say this species? Suppose mango plant is there. Varieties of mango plant is there. All are naturally not structurally compatible. Even they are same genus. How could you say that? How they are reproducing incompatible? So you have to think about also other markers. So they found those are not reproductively incompatible, but it changes in the secondary structure too. They found one CBC in line 3 or 2 or 3 CBC in 1 or 2 region. They found interesting they are totally reproductively incompatible. And particularly algae looks alike. So saman dekha some are sexual because sexual karandi. Apar kimti kore varieties form. You have to think another markers, and that marker gives this supplements and very concrete conclusions. If there are any changes found in your DNA, particularly in ITS2, they are incompatible.
very nice one.
Thank you, sir.
Before going to speak something, at least I give some brief introduction regarding that. Perhaps, কিন্তু সেভেন হচ্ছে ব্যক্তিত্ব 1989 সমস্ত উদ্দেশ্যটা টপ 10 ভিতরে ম্যাটেরিয়াল আছে এবং he of course was considered top 10 position, top 7 position in 1989 actually. সমস্ত আলোচনা সফল হয় বিজয়ী কিংবা রেনেসান্স করি সমস্ত আলো বিজ্ঞান পরিকাঠামো নেই কিন্তু বিজয়ী বলে তাদের সত্যবাদী ধর্মের মনে আদি মনে হচ্ছে তাদের সত্যবাদী ধর্ম দেওয়া our production scientist of the year by National Environmental Science Academy. You are awarded scientist of the year in the year 2020. And he has been awarded by the Odisha Botanical Society for significant contribution to the field of botany. And recently submitted a gene sequence to NCPI gene bank. Thank you. We are lucky enough to get such a type of scholar here, professor here. Okay, thank you.
Good afternoon. Thank you, sir, for your nice work. My esteemed teacher of all teachers, Professor Koraput. I request the entire house for a standing ovation for Professor Koraput because no one is better than Professor Koraput. And we respect him and I don't have any hesitation that he is a father of the soil. I belong to a very small village, and he belongs to another small village which is just nearby, and that is Jeypore. And I learned all those things, all those dedication, hard work from Professor Koraput, where he is from the same place, that is the son of the universe, Jeypore, whose words are written in UN, that he thought for universe, not for the art. The word Jagat that implies universe, and we both belong to the same soil. So I am thankful to the organizers, my esteemed teacher, Professor Koraput, Professor Sahu, Sanjay Kumar Sahu from Korean principal, most respected Dr. Hagar, my elder brother, my elder brother, and most talented, youngest scientist. I am not a big fellow. He is a big fellow. A big thank for Dr. Sujet Bhutia. Academic administration is alumni of our university, from our department, and the organization, Dr. Jena, Dr. Nayak, giving me this opportunity to speak a few words about my journey in Utkal, at Art Utkal. My guide, Professor Vikram, I did a toughest one. And when I joined the department, he denied me to use this time. The achievement you fetch for your identity. You are not allowed anymore in my lab. This was my first day joining, that means our. On that day, I thought that Professor Vikram is so selfish that he is not allowing this lab. But I did my PhD. You know, Goldsmith only, he knows how to make a sign out of the gold. That means all those impurities can be taken out if you put them in acid or put them in flame. Otherwise, you cannot. If I could have used his lab, might have this new, that means the research, what I did, in my opinion. Let's house design. That is what biocomposite is. And this idea by teaching students regarding cell wall, the fiber and matrix composition, how this cellulose fiber, whether embedded in matrix, can some fibers, natural fibers, be embedded with some natural matrix or biodegradable matrix, so that can that be the replacement for plastic? Why? Next slide. Next slide. So you see, these are the tips for World Environment Day. Now, in 2023, it will be by ecosystem restoration and all, the continuity of that. So you see, in 2022, only one art, ecosystem restoration, time for nature, with air pollution, with plastic pollution. That is what in 2018. And the year when I was awarded with a project from DBT, by fighting with all those experts, that biotechnology means everybody knows sequencing, cloning, transgenic, expression of gene, overexpression, inhibition, all these. Recombinant DNA technology. But I submitted the project regarding biocomposite to DBT, and all those expert committee, they denied me that this cannot be your project. We cannot consider your project. And luckily, Secretary DBT, he was there, and he was one administrator. But he could recognize me instantly. I said, biotechnology means that technology which should be transferred into the field. Not that I did my PhD in transgenic, agricultural, medical, genetic transformation, long back, probably the past thesis from Odisha to use efficiency. But I am submitting this, knowing biology, knowing biotechnology, but I want to do something for nature, something for the society. Maybe a very small one. If you give me money, then it's fine. If you don't give me, you cannot stop me to do money. So my objective to mitigate plastic pollution. Why? Because can we lead a life without plastic? When you wake up in the morning, starting from using the toothbrush, combing your hair, till you sleep, each and every, all those activities that needs plastic. And this plastic for the synthesis, they require very fundamental rules. Chemistry, what the degradation then to billions years. So that came to mind, and I submitted this project, awarded with this project, and one of those research fellows is now working, he's yet to obtain the thesis. Next slide, please. So what is this composite? It's a multi-phase that is for combination of natural fiber with some matrix. As I have suggested, I have chosen polylactic acid as the matrix because polylactic acid, that is what biodegradable, and I choose cylindrical because we can use wire, the coconut fiber. Similarly, Lupa cylindrical. Then the idea, this was also very interesting. Why Lupa? Because there is not there, but by roaming around with dotted and white, the stropping we need Lupa. That is made up of again plastic. But we have all those, that means in this district particularly or in Sambalpur, I have stayed, Pitta Corda. So that is what used as what Lupa for cleaning, what you know, the pedicure and manicure, all those things you can say. That is what, that drag material that was used. So that fiber that is what very, it has very, that means it's very resilient, tensility is high, it is resisting the stress and strain. So I thought the plan that how can I use this fiber with this matrix so that this may be the replacement for plastic? Can it be biodegradable? So this was the idea. Got the project? We started all the work again. Hurdle scale. That means when you don't have any name and fame, that means you are in the initial days of your struggle and start your career, then nobody is want to take you as a collaborator. Now I have, that means you won't believe, I have 5 collaborators that are approaching me. For this collaboration and everything. But on that point, I just do, that means Institute of Physics, even RNN just nearby. But I got the, that is help from Cancer Research Institute, Hemolata Cancer Research Institute, private hospital. Let me tell you all of these in detail. So this is what composite, no need to read all these things. The idea came that cell wall, like cell wall, we can go for, that is composite plastic synthesis or not. Next slide please. So this is what preparation of biocomposite. Here we have to extract the fiber and we have to mix it with what? Polylactic acid because both are biodegradable. That was the idea. Next. So you see, I was talking about this group. This is what Lupa. Next one. Next slide please. So you see, we have collected the samples because biotechnology, as we have suggested, sir, as this talk was specifically for sir, Professor Prabhat Mahapatra. So he knows all those things in detail. Sir, for DPT sake, I have collected the samples from different places and going forward, DNA fingerprinting. Is there any variation in fiber or not? This is one aspect I have to keep as per the, that means suggestion of the sir expert. Otherwise, I should not keep this.
No, that will be definitely various.
Yes. There is.
I've seen them in
Yes,
Japan. That what they do is there will be cracks on your
Yes. And they brush. They remove. Yes. Sir, actually my wife, she was using this plastic. So again quality standard. So why don't you use this one? Then I thought of this.
Very nice.
Next one. So you see, you can get this one lifa like this. You can get the extract and you can dry the fruit. So this that means whatever I am telling to all my young student friends, because research can be start with zero. No need to in a hurry that I have to do something. So with zero investment, with collaboration, with physics and chemistry, you can do wonders. I'm telling you. Just a minute. So this fruit that belongs to what? That is every district is a Borisa, lignocellulosic material, highly tensile and very strength is high as suggested. Scrubbing was used. Next one. So you see here actually scientific process starts. That is to begin the isolation of these fibers. We have to go for what? Acid and alkali treatment. So while using acid and alkali treatment, you see we have used procedures. That is, we have collected the fibers, dried it, then we kept it in oven, more dry it, then we treat with alkali and acids. But at the end, what we observe that to extract fiber for making composite becomes cost effective. So research should not be very costly. Research should be minimized. Minimize effort, minimize cost, and output should be maximum. In my opinion, one may accept it or deny it. Next one. So we have prepared this, that is composite material by taking PLA and all these fibers. So here we have that is these codons are for what? Composite material depending on their percentage of what? Polylactic acid and fiber content, and how that is the strengthenability can be that means more or less that we have to evaluate, and we did by making this formulation, and we have taken the help of CPET. At this level we got the help from CPET. So in this molded resin they are allowed, and we have prepared this FC fiber, that means leukocytic fiber along with all this PLA. So net, that means neat PLA, that means I say this is my standard material which is completely biodegradable, one fiber that is what completely biodegradable. So these codons are on the basis of what? The perfect. Next one. So we have tested this biodegradability. You won't believe we have not, that means degraded. That means by taking some hi-fi instrument, incubator, this, that, only soil, sand, compost, and by inoculating some fungal spores and bacterial spores, those are available in our lab very easily without investing a single part. So next one. So, but we have changed the environment, we have changed the temperature, we have changed everything. So where the degradation is maximum, and you won't believe the research lab, she came with the that means result. So this is not degrading. So this sample is degrading, but this is not degrading. But she is not. That means in a process to study my mind, I have having all those two different aspects of this research, because failure is a pillar of success. If it is not biodegradable, then I am going to be successful in a manner that if it is biodegradable, I may use it for some other purpose. If it is not biodegradable, I go for further study. Can it be applied in biomedical field for the preparation of artificial limb, artificial bone? So this was the idea. That means we have prepared the composite materials in a manner. Some will be degraded fast, some will not be degraded. So the research collab, she came with all those data tables, and she is telling that it is not degraded. I said it will not degrade. I have patience. Next one, please. So these are what the degradation by bacteria. We have got the result in bacillus substitution. That is what inoculum most effective. After every 15 days interval, we used to take the measurements only by measuring your normal weighing machine, not that any costly machine. Next one. Then we got Fusarium oxysporum fungus. That is what individually degrading fast. Next one. So you see, if that is not degraded, or if it is degraded. If it is degraded, this can be prepared, or these things can be prepared by those materials. If it is not degraded, next one, next one, next one, next one. So if it is not degraded, can it be possible to go for what artificial limb, bone, cartilage? So that was the idea. That is inside our body, they should remain forever. They should not be degraded. So that was the idea. Next one. So what happened? Again, to defend the first year report, whatever I am telling according to the stories of the project. So they suggested that if you are going to use so much of consumable, so much of chemicals for degradation, for preparing this, then it is cost effective. Then we should go for alternatives. That means how to minimize the cost to prepare the fiber. Without treating them with chemicals, we adapt another technology. We try to expose this fiber to X-ray, gamma ray, photon beam, so that if the adhesive property can be enhanced without treating them with chemicals, cost can be minimized, and that was the ID. Next one. So you see, as I suggested, this is what completely biodegradable. This is available. Next one. So you see, this hydrogen bonding, that is what important for resin and fiber, and that can be achieved physically if you treat. Then with what? X-ray, gamma ray. And for this one, we have to take the help of Hemalayan Cancer Research Institute, and they have that means clearly that is formatized to do this work, and we published a very good paper on the results, and we are on the process of submitting another one. During this 4-5 days, I will submit another one. Next one. So we have modified the fiber as I suggested through chemically or through physical method. Next. So this is for chemical treatment, which is very costly. Next. So this is the new method through the irradiation of electron beams. That means the adhesive property can be enhanced. Next one. Next one. So we have also tried for proton beam. Next one. So this is for the gap. That is what previous experts, that is whatever the coconut or other fibers they have used the biocomposite, they have mostly extracted the fiber through chemical method, and only electron beam they have applied. But we are now trying for X-ray, photon beam, gamma, and all this. So we'll on the verge of submission of another paper, let it be accepted, then I will unfold everything. So we are trying for that, and we are successful in that. That this matrix that becomes more resilient. Next one. So next. So this is what novelty I have already spoken. Next. So this is what the total that means flow diagram. How to prepare entire thing. Next one. So already I have spoken. So we have tested through FDM, Indian electron micrographs. Next. We have that is compare all these things. Next. So we have also prepared jute composite. A similar method for Jeypore, we have also prepared jute, and this was done by my one MP student, Aparna, and he has now at PGT and he is not pursuing this one anymore. So this is for stopped right now, but the jute one that is for continuing. Next one, the same process. Next, this is also like the same composite material and degradation, and we have published. You see, this was first paper. That is very primitive results and we have published. And this is what paper is Willy by that MP student, and she is now not pursuing this jute composite one. Next one. So these are some now the previous one. So you see, this is the paper I published. That is chemosphere, and with impact factor I say 9 point something. So no, that is you see very simple experiment without ignoring much cost, and already it is being published. Next. So I'll take only 5 minutes. Hydrogen medicine, this is what another aspect I am doing right now at Uttar, and one of my PhD scholars is there, and in your college, your skill college, and he has done some of the work related to this. So this is what very easy technology. Sukhinda is what highly polluted with chromite mines. So how to develop the technology with plants without investment, that these mining sites may be cleaned up. So this automation that fetched me some beautiful papers, but now I am interested in real science application, not that getting the paper published. So next one. This is next. So this is what that is for chromium we can go for, and likewise somebody is now doing some post-processing aluminum and nickel. I have seen in the upstairs, so I'll advise them how to perform those experiments. Next. So you see, this is what the district which is mostly polluted through chromium and chromium hexavalent, that is what highly toxic carcinogenic. And if you grow, let's say, are we doing wonderful research in rice? Let's say we go for rice cultivation. On one variety of rice, let's say, resisting chromium toxicity. But if chromium gets into the grain, then because of amplification into the food chain, that will create havoc. So our aim and objective to prevent chromium from rice, that is what? Accumulation. So if you remember, my grandpa, he was a lawyer and he belongs to Kolkata district. And when I was in class 9 or 8,
along with him, I visited the field. What sir was doing, that was at that time. Actually, we plant some Madunga, that means Uraisa, Rupipagar, Nivara, all these wild rice. And we plant them at the, that means boundaries. And we make the field, that means what, little bit, that means what, you can say in height, raise the field at the middle and gradually maintain the slope. And if we can go for that principle, let it be in mine soil, water will flow down and all these mine leach and this Madunga, that might take, because these are monopods and they will take all these fiber roots, take this, what you can say, toxic metals, and maybe our rice will be protected. And you won't believe, simple this design, we tried with lemongrass and only this concept with the model, that means only calculation, no modeling, that means no supercomputing, only some equations, and we got wonderful papers in chemistry. We have published 4 papers continuously, one after another, doing by this. Next one, next one, next one. This thing everybody knows. Next, because this two previous. You see, low level of chromium we have tested because I have interest, and these things are done by our MSc student during their cytology practicals only. While giving them onion root, I have given them the, that is this chromium treatment and nothing. And we get all these wonderful results, that is without investment, without spending money. That is I got all these pictures. Next. And chromium is responsible for what? All these lanyards and all those. Next one. So you see, previous one. You see, zinc oxide, nanocomposite. Again, some of our speakers they are speaking about silver nanocomposite. Silver nanocomposite, that is what very good antibacterial microbial property. And right now, these are being used in refrigerator, in AC, where nanodeodorizer, you can say this material that is what used. That is in nano form. If these metals they can create, that means this havoc, they can inhibit the pathogenic bacteria. That means these are highly toxic for our cell. But I did not have the opportunity to study at cellular level, at molecular level, because of that means we have been handicapped in the state university with all this. So next one. So this is from your Rayagada district, from Koraput. That is so. That is I was from the first time visiting this Jeypore, but one of our students, she was working at Jeypore paper mill, and this paper was that is published on this one. You can say hydrometallurgy, macrophytes, by using macrophytes, how to remediate this Jeypore paper mill effluent. Next. So you see, that is. We isolate some bacteria from chromium enriched soil as well as acid enriched soil. We have just submitted the sequences and all those things. Yet to receive all those details right now. Next one. Next. So this is for simple procedure. For simple procedure, you see, chromium toxicity, that's what highly significant. So we have tried our level best to mitigate this chromium toxicity. This is that is not related to my talk, for which I am invited here, that is biocomposite, but this one I am telling whatever we did in our physical during this last 2 years. Next. So these are the papers we have published out of this better toxicity. Next one. So next. So you see, previous one. You see, simple that is lemongrass calculation. Hemocarbide, he was that is doing all these things previously, but he was that is was our teacher. He was applying all those root the technologies, that is what only antioxidant enzymes and all, and we have that means added just nothing, that means microorganism, and with that some calculation, and we have added some that is what parameters whether oil content is changing or not. If oil content is not changing, then mitigating chromium toxicity by lemongrass will be useful, because oil never gets into our food chain, and lemongrass oil that is for other purpose. Because while publishing a paper, my students we got the yesterday only convocation. To publish that paper it becomes quite difficult, because we have taken macrodiagram, and once it is a food grain, reviewers ask for everything in bit where we fail to address many of those queries. Next one. That's all. Just a minute. Next one. So these are the papers we published on these simple experiments. So you see, this is what alleviating chromium toxicity in hot ground. Previous one, please. Previous one. So this is the study by lady over here. So she will explain in a much better way. So she has isolated some endobiotic bacteria, and these bacteria are highly resistant towards chromium. And while applying this strain to the chromium in this soil, this bacteria is taking all this chromium, protecting the plant, and plant is not getting the chromium toxicity, and grains are not affected. This was a study, and which was also recognized. Next one. So this is the entire protocol. Next. I will leave. So this is what we have submitted these bacteria and all sequence and everything. Next. So these are some prototypic parameters. I will explain in detail. I know less. So next. So these papers are actually this gives me pleasure and in front of Professor Prabhat Mahapatra to present this. So this was accepted this year. That means entire research work. That means the bacteria. Whatever we have isolated from chromium soil, we have submitted individually and synergetic effect. We have compared and we compared synergetic effect of both the bacteria. That is what most effective to mitigate chromium toxicity and protecting the plant entirely to get contaminated with chromium and get transplanted or get that is stored in the grains. So next one. So again, with the help of Sambalpur Vandana Guru, this is a very simple experiment we did with again mycorrhiza, and we have seen applying mycorrhiza again reduces chromium toxicity to a significant level. So this is another plant, Genia. We have tested because this is what flowering plant. So this is not edible. So no reviewer asks any question why you take this kind of plant in hydromedicine. Next one. So next one. So this is the ultimately paper. This guy is now continuing her PhD. He has published this paper during her MPhil. So next one. So this is what the outcome. You can reduce chromium load from cooking that without employing much investment with a simple technology by employing some plants, selecting some plants, planting or promoting the farmer to grow those plants in that area so that that area may be protected and can be reclamated for better cultivation. Next one. So thank you all. Though I have consumed much time before lunch, so I will excuse that my apology in front of you. They can watch about time before lunch, sir. And with your blessings and with your ideas, sir, that means physiology, sir, I learned from you. And whatever the papers I, that means in real sense, I appreciate Sambalpur University. That means they did wonderful work. No hesitation to declare here. And led by another professor, Professor Prabhat Mahapatra, whatever physiology I learned because we see, I am that means psychologist. I did my PhD in tissue culture. Because of my guides, that is what, inhibition, not to use his lab, makes me this kind of work. So, to sustain, you have to do something. And whatever I learned, I discussed with SARS many times, and I got many things from SARS paper. So, I owe all this publication to Professor Prabhat Mahapatra because of his, that is, all those publications I have seen, I have referred, and because of this, I am here creating my identity, at least at this. Thank you, sir.
I would not have spoken, but I am inducing me to speak. So this is really a commendable one, and really a commendable one because the publications he is showing, they are of high impact factors, and the idea, both the idea, he has spoken two things. One is the Lupa. The name is not coming into my mouth. So that is a very novel idea because I have seen abroad that I live in Japan or China several times. And these Japanese and Chinese, particularly the girls, most of the audience is girls. And they are very conscious of their toes, the heels, and the skin also. So they use these process. They made process. They have this, but they go for this one. And this is in high demand. So that is this subject. I never thought that Chinmoy will pick it up. It didn't come to my mind that a person will pick it up for his research studies. And that's why although he has already said that we are close neighbors by his habitat and my habitat become very close because I am from Jeypore and he is from the Ranapur side. So there is a close association was there previously. But in spite of that, I have no hesitation to tell that he is a genius. He is a genius.
All blessings, sir. So it's my humble submission to entire house for PG students I'm telling. So research you can do at your own place. Take collaboration with physics, mathematics, chemistry, statistics. Try to tag botany, zoology, physics, chemistry, and you will do wonderful papers like this. So there is no issue. I have not used that is you will say all these publications without investing much money because it's our taxpayers' money. So this is my humble submission to government also. So because I was refused in this project, that is entire panel refused me instantly, but they accepted and they gave me money. And thankful all my teachers, all my fellow colleagues, all the students, all the research scholars, and Uttal, and all the participants. Thank you. Thank you.
Thank you, Professor Pradhan. Thank you, Professor Jana. May I request Mrityunjay sir to come over here? I have a block for you. And I request Professor Koraput to come over and
I like the
now I invite Professor Sahu to come over. I request Professor Sahu to please come over.
Among the teacher because he that is very close to me because one of my close friends
on the presentation
Well, thank you very much, everyone. Now we have the best session of the seminar is going to be started. And in this session, I have three of our scholars, presentations. But most important is we are giving 5 minutes. Okay. So if you don't need anything to be submitted by our eminent professor, they can suggest you for your work. Try to avoid the result section of your work, not with your background, discussion and conclusion. That would be fine for us. Now I invite Dr. Sarani from please, sir, you here.
Please. Please have presence.
So, good afternoon, one and all.
This is your session.
Within a very glimpse, I have to finish as the time is quite very early. Myself, Dr. Usarani Brahma from Koraput University, representing something as already we have talked, less investment in our research and much more output from our experiment. So my topic is that last month I have delivered a talk about the technique about the sericulture. Now I am just going through some advances how we can manage within the adversity. Next please. So here is the introduction I have to give that it is quite eco-friendly, agro-business, cottage industry. We are having some more activities like horticulture. It is mainly belonging to the plant science, and in a composite manner, we have to gather something called rearing also. So rearing is the main part, which is mainly a lacuna in Koraput University. As Koraput University of Koraput is mainly based situated near the coastal region, mainly the temperature of climatic condition is quite adverse. Why? Because of the lack of proper marketization, storage facility, transport facility, due to the distance, price fluctuation, and so many lacunas are there. Koraput, we are all known that it is renowned as the Sin City of Odisha. But I'm so unfortunate that here only reading of Partosari is mainly done. None other than any kind of hearing till now has been done. But due to certain, as we have already discussed that government is not here to supply government job for one and every individual. So we have to overtake something for our own. So these are some scarce situation of our society. Next, unemployment. Next, women's slavery. Next, child labor. Old age ignorance. After the retirement, what they have to do. And the main one is my platforms, they are really moving to outside Lisa for Dadar. So, based on these things, I have done a very detailed work. It is not of a nanotechnology or a rocket science, only within a very minimum and investment. I try to give my best to my society for the poor and needy, as this is totally pollution free and domestic work. So next please, next. So many a time we have to motivate the rural people. Many a time. That's why I have taken the area for my investigation or experimental photos for Ganjam, some rural area, Ganjam and Gajpati. So sometimes we have to move. Apart from, I have given service now also continuing as a geology lecturer since 21 years. But apart from that, I am doing all such things for the needy. About the rearing process, because we are handling the living stocks. So life matters a lot, and it is just we have to handle with care because these are the works in the land. To work. Yes, I'm coming. Discuss your important. Yes, I'm coming. Next, please. So these are the things what we are going to discuss about the ongoing levels. Next. So about the life cycle, it is having the life cycle from the larva to growth. We are showing the charts and the lifetime. About the egg from egglets to the emergence of the mold, we are going among them to just give the knowledge and the techniques how to handle. Next, please. So these are the real works, not from an internet or something like that. Once upon a time, it has been believed that it was totally new. It is totally impossible. No one can do the work of rearing outside the hometown of Koraput, but myself, I did this, and these are the real things. Next, please, these are the lands, and in my home, within the 500 square meter area, I did this. Within a single room, I did this. Next, please. So by only by maintaining the temperature and humidity, I did this is my hand where the worms are mainly there. These are the eating of cocoons. So in this way, we get to the pupa stage. Now the imago. Yes, this is the chocolate already. And another one is just now I told that it is a part of the composite thing of geology. This is my backyard of my home garden where I have planted the mulberry. It is so nicely growing inside my hometown. Next. So here is the result. Better qualities of leaves as well as the worms. They can be done by feeding the vitamin C as extra nutrient. So these are the findings. In case of vitamin C, please, here is the diagram. The red color indication marker is the vitamin C, the worms treated with the vitamin C, showing the excellent result. So here we have taken SK6, SK7, the trait, resistant trait, where the plant weight. We are also saying next. We are also saying when we are talking about the hemoglobin protein, we are also saying the vitamin C is yielding more. Next. Here is the plant protein. We are also saying the vitamin C yielding the much more. Next. Here also the PSI is somatic index. showing the maximum yield in case of the worms paired with vitamin C. Next. Now, cocoon milk also it is very nice with their growing. So thank you. Next. So these are the some of my appreciations from the community of scientists where I got the best award from NICER. So next. Oral presentation. I got the best. Next. So it is the Indian Express edition, World Indian Express edition, where I got the women in science. So the students had horticulture, botany cultivation, along with sericulture, field engineering. Next. They took my work and got the first award. Next. All India Radio, Akashvani, Vision. Next. Next. Indian Express again. Next. From wherever. Next. So with my some delegates. Next. Niger achievement. Next. So thanks.
Only one question, if there is any, or suggestion from senior professors available. Only one.
Total the work is mainly dedicated towards the poor, for the people, poor and by the people.
Can it be possible that the rest of whatever that is you are telling, that the rest of silk logo is there, na? All these silk sarees, there is silk logo, madam. So, why don't you go over this applying geotagging of this Koraput restaurant? Whatever you are
Already by Pato, the sari living from Koraput got the GI tag.
Madam, I am telling you, you did your work, na? Try to submit it and
Yes,
try
sir.
to get it geotagged, so no one else can get this one.
Yes,
So that
thank you for your suggestion. Thank you, I will do that. Now, this also is going to handle this work, and we have talked to IAS Mr. Dr. Pandyan to tackle.
Very much, ma'am. In the lunch hour, if there is anything coming in the mind of students, they can speak. Dr. Pranam. Now I will request Vishnu Prasad Behra to make his presentation. I shall tell you again, this is for 5 minutes. If you exceed that, because we are evaluating it, we are going to make a best presenter presentation. Okay, award. So, like the competition, you have to stick to time.
Good afternoon, everyone. I am Mr. Vikram from United Order. I am going to present my work maintaining mitochondrial health to mitochondrial biogenesis and mitochondrial oral health. First of all, I think mitochondria is the parts of the cell. So apart from producing energy, it is also involved in other cellular activities like stem cell maintenance, and opposite cell health. So it is important to maintain the mitochondrial health. For that, it does so in the cell by dysfunctional mitochondria leads to various disease conditions, including cancer, by promoting factors like hormonal cross production and activating composite pathways like PHA pathway and several stress response pathway like the NR2 pathway, which helps in the survival of cancer cells in. So, the mitochondrial quality control is essential to maintain the mitochondrial health, and this is achieved by mitochondrial biogenesis, where the factors like SID1 activates PGC1 that leads to generation of new healthy mitochondria to maintain the mitochondrial pool inside the cell, and along with mitochondrial fission by the help of proteins like ALP1, which helps in segregating dysfunctional part of the mitochondria through a mechanism called as mitochondria by the help of proteins like P1 and PARP. Next, mitochondrial fusion. Here, mitochondrial fusion occurs to maintain the mitochondrial architecture inside the cell to maintain the energy homeostasis. So for our work, then chosen one small natural molecule activity of butane, and from it was from SI1 from it was from butane to that molecule, and we carried out our research with this molecule. Butane is commonly found in Odisha, in Butane Mahasuma, in Uday is called as Podasa. First we did an MTT assay and the IC50 value of butane, which came around 20 micromolar. Next we studied the anticoagulability effect of butane by doing colony assay and scratch assay. In both the assays we found the cells were unable to provide higher concentration of butane like 10, 15 and 20 micromolar. And so as we are interested in the status of micromolar health, so for our further experiments we chose smaller doses of butane, that is 1, 5 and 10 micromolar. Next we from Western world analysis, we study the expression of SI1 as I said that is the SI1 molecule activating molecule. So first we study the expression of SI1 and we found a dose different increase in the expression of SI1. Then we examine another factor for mitochondrial biogenesis, that is COX-7. Similar to SI1 we also get a dose different increase in the expression of COX-7, a dose different of butane. This data was validated by immunofluorescence assay by taking compound as a marker. We also found an increase in intensity of property. This leads to the conclusion that butane promotes mitochondrial increasing number. Whether these increasing number are functional or dysfunctional, we did post-cytokine assay by taking gas like MTs and MTR, which and the first thing in MTs and MTR removes the butane in turn activates healthy mitochondrial population. Again, another factor of mitochondria, that is the mitochondrial membrane potential, whether butane is disrupting it or not, or that will be increasing with fluorescence emission by taking rhodamine 123, and we did not see any change in this infer that butane maintains the mitochondrial membrane potential. Again, we cross-related this data with by taking one known disruptor of mitochondrial membrane potential, that is delicolin. And as you can see here, in case of delicolin, we found the increase in the intensity of rhodamine 123, but it is negative during the combination treatment of delicolin and butane. Similarly, ROS is also another aspect of mitochondria. Here also we take positive control delicolin by taking GH as the type for calculating the mitochondrial ROS. In case of, as you can see, in case of delicolin, we found the increase in intensity of GH, but it is negative in presence of butane. All these data were again cross-verified by taking post-cytokinetic analysis. Again, next we check for another factors, key factors that are responsible for mitochondrial biogenesis. Here we take PCC1 alpha, which is a known mitochondrial biogenesis factor. Here we found a dose increase in PCC1 alpha. We cross-verified it by taking the inhibitor of PCC1 alpha, that is SR18292. We got similar results in our immunofluorescence assay by taking SR18292 and SR1 approach of PCC1 alpha. In all the cases, we found that butane can restore the lost effect of PCC1 inhibitor. Next, we check for another axis, that is SI1 PCC1 axis, by taking the inhibitor of SI1, that is NBX527. And here you can see in presence of the inhibitor, we can see the excess of PCC1 alpha, similarly COX-7. But combinatorial treatment, we found restored expression of all these, and these were also verified in by immunofluorescence and SIR approach. Next, we check whether mitophagy is essential for maintaining the mitochondrial health. So we check the effect of butane on mitophagy. We also found that butane can promote mitophagy. So this is the summary of my presentation. In lower dose of butane, we see that it maintains the mitochondrial health through mitochondrial biogenesis, mitophagy, increasing loss production, which in turn disrupts the mitochondrial dysfunction and accept of your mitochondria, which in turn hinders the cancer progression. But at higher doses, butane promotes apoptosis to check cancer progression. I'd like to acknowledge my mentor, Dr. Sujit Kumar Gujral, and this is our team. NIT Rockero for providing infrastructure, especially for fellowship. Thank you.
Thank you very much. We go Vishnu. So if there is
there will be a single question.
By maintaining the mitochondrial health. For releasing preliminary can be done or not, you have to be another for cancer only. Here we have seen only mitochondrial aspect. For other place, you have to look into cancer perspective. It is not cancer patient, but you have to do all those things.
Next presenter will be one of my students, Dilraj. Dilraj Pawar. I was a teacher over here. Next presentation will be by Vikram.
Good afternoon, everyone.
Please come and give
Respected teachers, students, research scholars from all the different universities and colleges. Today I am going to give a talk on systematics and molecular characterization of fishes from Mahanadi River of Western Odisha. These are the different contents. As you can see, I am not going to elaborate here because the time is short. Introduction. Mahanadi River, as you all know, that it has a large basin and also it extends from 19 degrees 20 minutes to 23 degrees 35 minutes towards north and 80 degrees 30 minutes to 86 degrees 15 minutes towards east. And it originates from Dhamkari district. This is the status of different fish diversities of Mahanadi River. Earlier, these all people in different years, they have reported this. The study area, we have taken Western Odisha, it has 10 districts, out of which we have focused on 3 districts. That is Bargarh, Balangir and Subarnapur. These are the 2 objectives we have taken, that is systematics and biodiversity study and the DNA barcoding using the COI gene sequence. Materials and methods, we have collected all the species of fishes from different areas in different seasons and we have taken them for DNA analysis. First we have kept them in formalin and then we have taken for tissue studies or DNA studies in ethanol. And we have also studied or named all the species according to Jeypore and we have referred all the different papers, children. These are the different species of fishes which are found in this area. Some of the fishes are yet to be identified. And as you have seen, the latest studies, out of so many fishes earlier reported, we have reported this year, these 11 new species we have reported or we have studied. And as you can see in the pie chart, Cyprinidae family is showing the highest number of fishes out of our study. Genetic analysis of the fish step. First, we have taken the tissue samples and we have used the sorting out method. We have taken the primers, step 1 and R1, that is forward and reverse primers. Then we have done the COI gene amplification by using the Sen et al. And then et al. 2019 papers. These are the different species whose accession number we have already got. These are 2 interesting findings in that area. First one is Leodon mutia. This is a copper fish. It normally lives in saline conditions. But how it has migrated to the freshwater fish or freshwater conditions, we are studying about it. And second one is Stereoliptic Cardalis. This is a sucker mouth fish and it is an alien invasive species. But again, we don't know what is the exact condition, how they have come to the freshwater condition, and whether it has any negative impacts on the native species. These are the different results I have already discussed. We have collected 14 new types of species. Out of these, 23 families are reported and 12 species are reported for the first time in Mahanadi River. And gene sequencing of 32 species is already done and circulated. I have already discussed. The significance of the study, you will see here, identification with novel species. Systematic studies and genetic analysis, socio-economic development of the people of that area because they are included backward. Most of the time people think that they are backward, but it is not so. Yes. Focus on ornamental fishes, indigenous species, and job creation. We want to make a thematic presentation, and we also want to give a baseline information of what are the different species which is present in that area, whether it is harmful or whether this is helping the native species to develop them. Thank you very much. These are the different differences.
Thank you, Vikram. Now, one question or one suggestion. Told that you have what's your name? Where you are working?
I'm working in Punjab
For whom? Oh, as a teacher. Very teacher. Yes. Barabar. My question is that you have import the pieces from the district. Yes or no? One in Barabar and another is Jeypore and Khari Subur. Bolagi? And you said that you have identified 12 new species. Yes, that's why it is called new species. Yes. Yes. This species are first time we have reported. Correct? Again, it is not reported. Yes or no? Are you understanding my point? You told that for the first time you have reported 12 species, 12 species which were not reported earlier.
which was not earlier studied.
Studied. Not reported. Yes, let me. These 12 species you reported in districts they are available or it is confined to one or it may be available in Barabar, it may not be available in Baladi, or it may not be available in Koraput, or like that. Second question is that there is a lot of hue and cry nowadays because of the industries. Those are in the vicinity of Mahanadi Bank and Reservoir. The diversity of everything, including the fish, is declining. Yes. And at this stage, we are claiming that we have the new species we have identified and which have not been studied earlier or reported earlier. So this is most important.
And one of the things that I want to cite here, it's also blasting to take the species. Because of this, the diversity is decreasing.
But at the same time, we are telling that is you see, industry, because of industrialization, a lot of industrial food adaptation and all that, and all the diversity, including algal biodiversity and many other biodiversity, plankton, all are decreasing. This diversity also we have paper that it is decreasing. So you have to rethink and verify your data.
Thank you, Professor Sahu. Thank you, Jeypore. There are many things we can suggest during lunch. Discuss with sir. Simple suggestions and knowledge teacher. Now I shall invite Chandra Hasini. Dynamic.
Chandra Hasini. Very good name. Chandra Hasini is a place in Chhattisgarh. You know, a temple. Yeah.
There are five visitors there.
There is a water park there also. introduce yourself.
Blinded.
Please go to your objective.
Their current changes their background. The panic and darkening of the blinded pieces. The harm of melancholy in the panic of the blinded. Or making him sit around their eyeball. The correct changes. Was prepared. After driving, the pieces suddenly changed to their dark color because of the but after some time, some other subjects are there who keep the melancholy and suddenly they started wearing their parents. Some family table, the rate of panic was done because some other like red are there. They release the melanin. Decrease of melanin causes the red of color. Black color shows the melanin secretion of the melanin pigment around their skin. So the fraction of the melanin last minute, the 100% annihilation was observed. This is the source of panic of the receptor. The panic of the receptor was shown the natural color of the and we can see through the color line is just after it. The rate of panic was because some other places release the melatonin. Then we inject the melatonin solution to the of the receptor. That the visualizer changes changes of the pieces to the background adaptation is a physiological process. But it's just not a physiological process, but it's also a psychological process. Because there are not only the photoreceptor organ like eyes are released to melatonin, but some other organs like DHH and the enzyme acetyl transferase enzyme are there which release the melatonin. Because of that, the if we remove their eyes, the rate of panic was observed. Phytolytic process in this first is the fast and the second one is there are many pollution factors which cause the aquatic pollution that may affect their eyes. And if the eye was affected, they already affect their physiological changes. And if the physiological changes were affected, and they are highly dependent on the sources for the protein consumption, then the ultimate
Thank you, dear.
You have come all the way last week to make this presentation. Good. That's good one. So one suggestion on the hello. That is the title of the work. Blinded. What is the findings in one sentence? Important findings. हाँ।
सेकेंडरी डार्क कलर में चेंज आ रहा है क्योंकि उनकी बॉडी में उनका तो आई होता है प्रोडक्ट अगर ऑर्डर आई को नहीं रिमूव कर देते हैं तो सीनो को हम खाता तो मैं
Thank you. Very much everyone. How we invite you for running lines. It was a report that here is RP at 2:45. Students' presentations will continue by 2:45. So whether someone is there or not, evaluated. So now, may I request everyone to just be present for 5 minutes? Just to leave for Koraput. So he will make his presentation for 5 minutes, then he will.
Thank you, jury member, esteemed chairperson and co-chairperson, my teacher and my student and my dedicated friends also. Myself, Dr. Samendra Narayan Malik, working as the assistant professor RI Bhubaneswar, will deliver on the topic foods used by the ethnic people of the Sundarbad youth. And I will talk on the uses only of the food as a wild food, because many uses are used by the foods by the ethnic people. Next. Generally, woods. When we listen to woods, we think that that is the cannabis sativa. That is the woods are using the narcotics, but that is not the thing. That the woods are using plants grown in the wrong places. So generally, they are the herbaceous and not useful much more. That's why that is the important thing we have to study during the floral part and the technical study to the preparation of the floral study of the Sundarbad district. I thought this should be studied because this things should be useful in the future. Next. As I have told that the woods is the wild food. Wild food, what? That means that not used but the domesticated, not cultivated, but grown and collected from the environment. Here and there, they are growing this very automatically naturally. From there, we are collecting with their food. These are my objectives. Study diversity. The documentation of indigenous in traditional, because in the 95th edition of the Mann Ki Baat by Dr. Prime Minister, they told about the study about the traditional knowledge. So I have taken this, the documentation of these tools as a first part. Last, the proximate analysis to evaluate the ecological knowledge with our nature, our science. We have visited different places of Sundarbad, documented with the tribal peoples present there. Out of the tribes present in Odisha, 62 tribes are present here. But in the Sundarbad, there are 40 tribes are present, out of which the Munda, Orang, Gond, Kisan, and Kharia Bhais are the primitive tribal group. The 30 primitive tribal groups are present in Sundarbad, and we have visited these villages in the Sundarbad district and for the study of this. And here we have studied different areas to get the ideas and ecology of the studies for the food as well as not only the foods, they use the different other than foods. Out of the study about the diversity of the woods, we collected 180 species of woods. Out of which dichotomies are having more 74%, 19%, and these 54 species plants are used as the food. That means from the woods we are getting the food, and and then collectively by the tribal people, not only the staple people. Get the result. We have found that the most of the woods are the dicotyledons, not the monocotyledons, because the monocotyledons are used for their for the fodder plants or for the full purposes. But the dicots are the 85% are using for the our food purpose from the woods. I was the and these are the results. You can see the number of vegetables are found from the woods in the markets also. And here we can see from the woods they are getting the economy also. From their woods they are the selling and getting the that life substances for the daily uses by better process by the selling in the market local hats also. And the our quantity are they using by the daily wise. Most of the plants which are sold in the market. Next. Also to evaluate the our study about the why they are using as a food. Study about the nutritional process, the nutritional and antinutritional properties. And they show the good properties then that our staple plants, which are using daily life also, but these are the good sources also. That's why this is a very much good study for the future uses. Conclusion we find that this plant can be the another protein and during the hunger we can use it there because it shows the number of nutritional values and higher than the our staple. There is no need of any cultivation also there. We should only the domesticate. As the Sundargarh district is the full of all their industries, steel industries is there. So that's why the climate change in global structure there or the global warming adaptation should be there by the wood species also. And the younger generation are the moving away from the indigenous knowledge and conservation. So by the documentation of this structure and the study about this wood by the floral study, we can document the plants and the uses in the future and as well as we can assessment the biodiversity. What number of bodies are present now, what was the there and will be present in the future due to the industrialization. So that's the should be studied there. And last, it will be helpful for the completion of the flora because I am working on the flora because it is not completed yet. So I will be trying to my best to complete this flora. Thank you. I am giving the suggestions in what I am doing. Forest as word is one mirror reflection. As what we do the forest, we will get that then reflection. And in addition to this, another thing I will say during the study by the different groups of our committee or the members of our study, we found one wood species from the Jeypore, that is the Salvia misela, by one of our study group. That is the also another wild group, and we are working on the medicinal as well as the evaluation of the oil plants because it's the lamiaceae plant. So we are studying on the other the oil study. Thank you.
Just one clarification.
Yes.
We have studied the quiz that is available in Sundargarh district and Jeypore.
Yes, sir.
Yes. My question is that in your any slide I did not find this. You might have done that.
Yes,
and my question is that you see there are different parts of the wood.
Yes.
Am I correct?
Yes.
It may be leaf, it may be the
Yes.
flower, it may be the root. So
Yes,
which part is there? Have you identified these parts?
Leafy parts are
Only leafy parts.
I mentioned
Right.
the
Have you
focus,
studied the other parts? Root may be used
Yes,
for
sir,
the
but
food or stem may be used.
states are only in the monocrop
Yes.
leaves.
That
Okay, I have
bifurcation.
also shown that
My
in the-
suggestion is that
Yes, sir.
you please see that bifurcation.
Okay, sir, I will do.
And you see which part is more nutritious
Yes,
and
sir.
whether that is palatable or not.
Yes,
That
sir.
is my suggestion.
What is your study site?
Morning visit in Laukela and now studying myself in the It is my personal work, not by the our other teachers. And my personal name doing work.
So what is the justification of selecting Sundargarh district?
Sir, because there is a flora is not completed at first and the ethnoboty is completed. But during the flora study, I found that we should do it because my PhD work is in the wild food plants from the tribal belt. So that's why I related this plant, this study to this.
So you told that it is on the area of industry, only access to Laukela.